All right, so active versus passive transport is what we're looking at today. So with diffusion and osmosis, molecules move from high concentration to low, from where there's a lot to where there's a little. Eventually, they're going to reach what? What do they reach? Equilibrium, yeah. But sometimes this isn't the case. Sometimes we don't want them to be equal. What if we want to get a lot of sugar into the cell? We want to get more and more inside and less outside. Or what if we want to get more and more waste outside of the cell and not have a lot inside? Sometimes we don't always want to have them reach equilibrium. So the question then is, what happens if we need to move molecules the other way, from a low concentration to a high concentration? That's what we're going to talk about today. So we have a little table in our notes chart, and we are going to fill in that table. Passive transport. When we think of the word passive versus active, if I'm active, what does that mean? Active. Yeah, bye.
You're like
doing things, moving around, using energy. Passive is just kind of like go with the flow. The stuff we've been learning about, diffusion and osmosis, that's an example of passive transport. So we're going to fill in the left side of this chart with what we know already about diffusion and osmosis, because diffusion and osmosis are called this category of transport called passive. So anything that doesn't use energy to move molecules is an example of passive transport. We haven't talked about water needing to use energy to move. So osmosis and then diffusion, they don't need energy. They don't require energy in order to move. So no energy needed. We already know that they're going to go from a high concentration to a low concentration. So from a high concentration to a low concentration, where there's a lot to where there's a little, they want to reach equilibrium. Okay, you can go ahead and draw that little picture on the bottom if you want. There's some extra space. All right. So the other thing, we're going to write down our examples here. So our examples of passive transport include osmosis and diffusion, which we know about, but also facilitated diffusion, which we're going to talk about what the heck that is. So osmosis diffusion and facilitated diffusion. Yesterday, when we made a model of the cell membrane, some of the molecules, if they were really tiny, they could probably squeeze in between those toothpicks and marshmallows. Some of the molecules, like sugar, was really big. It couldn't fit through there. What did we need to make in our cell membrane for sugar to get through? The channel, the protein channels. We made pipe cleaners. So diffusion, facilitate. Facilitate means to help. It's diffusion, moving from high to low, but the molecules are big and they need to be helped by a channel protein. So we have to have a protein helping. So take a look at this picture. Simple diffusion is on the left. This is regular diffusion. The molecules can go through the cell membrane. They can fit through the toothpicks and the marshmallows. On the right, the molecules are too big and they need help to go through the cell membrane. So they use a channel. So facilitated, to facilitate means to help. You have this definition down below your chart, or the word down there. So you can write that: diffusion using a protein. So it's still diffusion. It's still going to go from high to low. It's not going to use energy, but this time it's going to use a protein. So regular diffusion is what we call simple diffusion. Facilitated diffusion just uses a protein. There's different types of proteins. You don't necessarily need to know the difference. One of them makes long skinny channels. The other has a little like shape for that molecule to fit into. Okay. All right. So now that we know that, we need to go through and talk about the other side of things. What happens when we don't want to get to equilibrium? We want to go pump as many molecules to one side of the cell. This is called active transport. As the name says, active, if I'm being active, I'm using a lot of energy. So active transport uses energy. So requires energy is the first thing. Yeah.
So if facilitated diffusion is like, they need like the receptors and stuff, does that mean molecules like naturally go to the receptors without energy?
Yeah. Mm-hmm. So we need energy. It will also move molecules from low to high concentration. Again, you can go ahead and draw this picture if that's going to be helpful for you. All right. So here is an illustration. It is showing ATP. That's the energy of the cell. Do you guys remember which organelle, which part of the cell makes energy? The mitochondria. You don't need to know the three letters ATP. We'll see them later on and stuff. But notice we're going from a low concentration on the left to a high concentration on the right. They're not going to be equal, okay? So what are some examples then? We have endocytosis and exocytosis. We haven't learned about those. We're going to take a look at those in a second. So if we look at the beginning, endocytosis, see the word in or N in front of it? That means stuff is going in. Exocytosis, stuff is exiting. So let's take a look here. So I have an animation. This is a paramecium. It's an amoeba. Amoeba is the big guy. He is wrapping his arms. His cell membrane is going to surround this little guy. So the cell membrane is going around so that it can bring this food, this organism, as food into its cell, okay, into its body. So this guy has now got him trapped. He's going to release some enzymes that'll kill this guy, and then that's his food. So this is one way that they can bring in big organisms or molecules as food. You can see him moving around there. Okay. So bringing something into our cell. The next one here is showing white blood cells. They're kind of the blob. The little ovals are the bacteria. The white blood cells are coming in and they're kind of surrounding and gobbling up the little bacteria pieces. So they're surrounding the bacteria, bringing them into the cell where they'll destroy them. As this video continues, we are going to see another amoeba here in a second. So this is another amoeba, and this one's trying to capture some food as well. And so this paramecium here is going to be its food. And so eventually this guy, the cell membrane is going to go all the way around. He's going to get trapped, and then they'll use him as food. So down below your chart, you have this word that you can write the definition for, endocytosis. Cell membrane surrounds an object. We don't really see the receptors in those videos. You can see them on the left-hand image there, the little Y-shaped receptors. But the cell membrane surrounds an object. It's got to be a big thing that can't fit through a channel. So a big thing, it's going to surround it and pull it into the cell. So let's look at an example here of the other one, exocytosis. So this is when something is exiting a cell. So watch this. This is an organism, and it's going to let go of things outside of the cell membrane, and they're big things, okay? What's really happening, I'm going to show you in a second, kind of like a close-up of what's actually going on. So look at this animation. See this little like circle full of stuff? Remember we talked about the Golgi body packaging stuff in these little circles, like proteins? They come and they merge with the cell membrane and then release all of their contents. So exocytosis is stuff exiting the cell. The vesicle is that little circle thing. So having that little circle, that little package of whatever it's carrying, whether it be waste or proteins, fusing with the cell membrane. And then if you want to draw a little picture, it's the exact opposite picture as the one you just drew. Still see pencils writing, so I'll wait a second here. So this is the picture you can draw then for that.